So it's a real pleasure to welcome you at the EuroBioC2022. So you can see here, this is Heidelberg in the sunshine. We've had a really long and dry and sunny summer, and for some reason the weather gods decided to switch to auto mode just today. So my apologies for that, but I'm sure we will have a fun and exciting meeting nevertheless, and maybe even with all of this. I would like to start the meeting with thanking the sponsors of the meeting. It wouldn't be possible to have such meetings, at least not for competitive participation rates, without our sponsors. So a lot of thanks to them. This is really appreciated. In the same way, there's a big team of people who have been thinking about this conference and organizing it and dealing with all of the various logistics aspects of the conference over the last months. So also a big thank you to all of to everybody here. It's been fantastic work, and obviously I hope we can keep this up for the next rounds. We have a code of conduct. The Bioconductor body is an open approach to science, and also sort of a diverse, an inclusive and respectful, polite way of interacting between us. So I think that's sort of self-evident. If there's any problems, there's a code of conduct committee, and you should contact one of the people named here. Some logistical remarks. All the information that you should need is on the conference website, because you're aware of the URL. Demos will take place more or less in the same floor, neighboring rooms. So the titles on the door, so it should be easy to find them. There's the opportunity to give flash talks for everybody who doesn't give a sort of an accepted talk yet. So you can look at this Google Sheet and subscribe your flash talk in the Google Sheet. Please go ahead. We have some spots of the feather sessions on Friday. So basically for picking up topics of interest, emerging topics, things that sort of are of interest to more than one person. So any suggestions are really welcome. And please just open a GitHub issue with suggestions of such sessions, and then we will have these on Friday. There's a live streaming, you're all aware of this, so links are available, and we have posters. So before the poster session, everybody that presents a poster can give a pitch of their poster, so that should hopefully enable you to get a quick overview of what's out there right away. So this meeting, in fact, has quite some history. I think we started somewhere around the mid-2000s in Lausanne with a number of European developer meetings, and then moved to Heidelberg in 2010. And then there was sort of so the idea of this meeting is to switch back and forth between different places in Europe, sort of where there's interested communities. So we were obviously Heidelberg 2010, we were switching back and forth between the UK and mainland Europe over the years. Zurich and Basel and Munich have been so very fun and hospital sites, also Manchester. The last in-person meeting was in Brussels 2019, which was a fantastic meeting. 2020, unfortunately, had to, for obvious reasons, had to be moved online. There was a heroic effort by the team led by Davide. And so now this is in fact a sort of belated instantiation of the 2021 in-person meeting. So in some way we merged 2021, 2022. And now we hope, of course, to keep this up again over the coming years and have an annual event that again moves around European places. We founded the society recently, sort of to help a little bit with the logistics aspects of this. I mean, in particular, to carry a little bit of funding between the events and also sort of organizational infrastructure to make it easier for this meeting to take place in different places and to help local organizers. So if you're interested in the society, you can talk to me or Simone or anybody. Yeah. So Bioconductor, in fact, started now 21 years ago at the time as a platform for analyzing microarray data. But I guess the bigger idea is that it's a platform for analyzing sort of large-scale, high-throughput biology data, and of course those challenges have remained. The technologies have been evolving over the years, and we've seen RNAs, we've seen ChIP-seq, tiling arrays, RNA-seq, single-cell sequencing, proteomics, microscopy, now spatial omics, increasing mass spectrometry. So you can see the technologies are getting more diverse, more exciting, obviously more highly resolved, and ever more voluminous and multimodal. And that's, of course, exactly the kind of challenges that we want to address, and what the project is good for. So it has sort of the main driving idea is that we have a collaborative approach to software development. So not just a team of people who write software and everybody else is supposed to use it. This is really more a collaborative platform with lots of interoperable modules where people can plug together things that they need, and also for sometimes maybe there's competing approaches, and then users can basically decide which one actually works best. Over the time, it has turned into a really big software repository. So we now have over 2,000 packages. That has its advantages and challenges in terms of maintenance cost, but it is also that. There's a lively support site for bioinformatics. There's now, I guess, more and more online fora where we can discuss questions about bioinformatics topics, and you're all very encouraged to engage in these. I think this can be extremely useful to have discussions about also thematical approaches on such sites, and then also to make them findable for everybody else. Bioconductor is like this, has only a very small team of a lot of people who do this as a job full time. There's about 6 people, mostly based in Boston. But then, of course, a lot of the actual content and the strength of the project also comes from the contributions from all over the world. If you ever wondered why there's a conductor in the name, I mean, that's the idea that you have lots of different experts, people who are really good in playing the violin or the cello or whatever they play. And then there's some kind of orchestration to make this all sound together in a useful, harmonic way. So there's 2 main sort of guiding principles in the project: the user experience, which maybe many people come for in the beginning, so that you should be able to find the best, scientifically most advanced methods for certain problems in the project, that the installation of these methods should be straightforward. And that's a big sort of topic to make it easy enough to install things. And it's a sort of big difference between things that are just on GitHub versus things that are on Bioconductor. Another thing is that we are keen to have good documentation with the packages, so that it should be easy enough for anybody new to discover what the package can do and how to plug together different workflows. At the same time, we also want to support an active open developer community. That's the collaborative development approach aspect. That's sort of the second phase or side of the project. In many cases, the people who really write the best methods tend to be scientists, biologists, or for other science. They don't necessarily have the best training in software development, but there's a bit of a tension, and we also try to help people to learn the good techniques of software development to overcome these hurdles and to turn their scientific expertise into decent software. So one of the key words in the project is interoperability. We don't want that somebody involved implements the big monolithic block that does everything. There's lots of different modules that are supposed to work together using exchange or interoperable data formats, data structures, and that's so the users can plug together what they need. The common data structures are really, even if they might be boring at some level, they're really, well, everything is built upon the foundation. Today we have over 2,000 packages, and we cover really the whole range of technologies, from sequencing-based assays, bulk, and now, of course, more and more single-cell-based, multimodal single-cell data. In some sense, everything that leads to count tables. there's now a very sort of strong dynamic movement towards employing spatial omics data. And of course, this is a whole new dimension of information that you can also see the single cell resolution, how tissues and organisms are sort of connected, spatially at the molecular level. So this is exciting. And then there's a number of other classics, flow cytometry, proteomics, metabolomics. And data integration is, of course, a growing a topic that we want of importance. One of the important themes is to enable reproducible research and open science. And that has a lot of implications. Also, I mean, a recurring theme with many of the things that we're doing is to sort of the pre-processing and quality control of new data types and interoperability. I mentioned this. They just sort of just represent the fact that the project is international and it's really happening all across the world. You can see here, I think, something like the site users per location. And you can see that the project is being used basically everywhere where biology is being done. And that there's also this aspect of bringing bioinformatics expertise to the world. How can you get more involved? So the first step certainly is to become a user, or maybe to learn and become an expert user at some level, join discussions and help others on the support side, contribute content. So this can obviously be packages, but there's also a big need and interest in workflows that plug together different packages and sort of provide know-how how to do things, even write book chapters or books, provide data sets. There's many different ways of contributing. At some level, there's also interest in people who can become package reviewers and help with that process because it's very laborious and but also very important for the project. There's different working groups that are trying to advance the project in different directions, and you can keep an eye on these on the URL mentioned here. Then there's at some level, there's a technical and community advisory boards that are open and that have a transparent selection or election process, and there's a yearly call for new members. So please look at these options and join in if you like. And of course, come to the conferences like this one, the workshops, and look at the event calendar. I think I said enough. So enjoy the conference, and I will pass to Charlotte, who will introduce Shila.
Are there any questions about what Wolfgang just said? Technical or logistical questions? Everyone knows where to go. I think we can start. We're a little bit early. Okay. Cool. So welcome also from my side. Let's remove these slides and open the right slides. Here. Excellent. So it's a great pleasure for me to open the scientific content of the meeting and to introduce Shila Ghazanfar, who is here to give our first keynote. So Shila is a research associate from the University of Sydney, where she started her group actually a few months ago. Before that, she was a postdoc in Cambridge with John Marioni. And Shila is working on developing statistical and computational methods for understanding cellular heterogeneity and interaction using kind of spatial genomics, and also integrating that with non-spatial single-cell data. And I believe that's what we will hear about today. So Shila, the stage is yours.
Hello, everybody. Let me just get myself organized. Wow. So I think just looking from the front seat here, I didn't realize how many people there are. It's amazing to see you all here in person, and it's wonderful to be here. Thank you so much to the organizing committee for the invitation. I really, really appreciate it, and I hope I can do justice to the wonderful content that I'm seeing on the program for the next few days. So I'm going to speak overall about a few projects from my lab, and then try to sort of expand out into, you know, what we're all here for in terms of Bioconductor. So with that, I'd like to give a bit of a background in terms of, you know, where I've been working for the last few years, and the type of biological questions that we've been interested in. So let me see if I can move my thing. That way? Yes. So during my postdoc, one of the key questions in the group was the study of early organogenesis in mice. And so one of the sort of key components here is, you know, along this time across these embryonic days, this work, 6.5 over to 8.5, quite a number of massive changes occur in a developing embryo. So you have sort of a very relatively simple structure of sort of 3 layers, and then a sort of explosion of different types of cell types emerging over this time. And so one of the key studies that came out of the lab was a study of single cell transcriptomes across the different time course. And so what I'm showing here is a UMAP representation that's changing over the time course. And yes, what we can see is that, you know, in terms of the various cell types that are emerging, we're seeing quite a number of different types come out across this time. But the thing to note is that, you know, this is a UMAP representation. This is our sort of reconstruction of types. And it doesn't necessarily correspond to what we're seeing in terms of the organization of these particular cells in space. And so that's where one of my key projects from my postdoc came in. So, you know, we were interested very much in understanding early organogenesis in mice, but in space. So here we collaborated with, so I sort of co-led this with a wonderful PhD student, now graduated, Tim Lohoff. And as part of the Wellcome Trust Mouse Gastrulation Consortium, we collaborated with Professor Long Kai, as well as a number of really wonderful people here to profile mouse early organogenesis at the single molecule resolution. So here what we're seeing is a sagittal section of a mouse embryo. So this part here corresponds to like the head. This is the heart area. Along the side is the sort of the developing spine, and then going around is the tail area. And so what you can see, this is a digital in situ sort of recapitulation of just a few sets of, just a few genes that we selected. So overall, we profiled some hundreds of genes, 350 genes. And what we're able to do is identify for these sets of genes where each molecule is located. And I guess I want to sort of highlight that, you know, this data set is quite multi-scale, right? So, you know, we have a whole organism section here, but if we zoom in, then what we're seeing is this at the tissue scale. But then if we zoom in even more, then, you know, we're capturing data very much at the subcellular resolution. And so, you know, that itself brings on challenges for analysis and opportunities, I guess, for biological discovery as well. So, what is this technology? In terms of what we did for the project itself, we collected 3 biological replicates of mouse embryos at this stage of early organogenesis, and we took these sagittal sections, so taking basically like a whole body cross section, and we subjected it to the SeqFish platform. So this is a technology where we perform serial hybridization followed by imaging to be able to identify these molecules in space. I mean, following on from that, there were a number of sort of imaging processing components, and then on to downstream computational analysis. So the SeqFish platform is highly multiplexed, and this multiplexing arrives basically via performing repeated rounds of hybridization as well as imaging. And so here we've got a schematic. So this, we can pretend this is a cell, that's a nucleus then. This is the sort of cell boundary. We've got the cytoplasm. Each of these little blobs correspond to molecules, so RNA molecules. And so then as we go through the hybridizations, what we have is that select molecules are fluorescing, and then we strip and then fluoresce once more, and then essentially we're collapsing this into a pseudo color image and then performing it again. Right, so the end result for us is that we essentially have a barcode system for the molecules to identify what gene it corresponds to. So that is the sort of data collection component, and what we have is essentially a set of images. So that's for the molecules themselves. But an additional component to this data collection is on the cell membrane as well. So what Tim did while he was visiting Long's lab was develop this additional component where, we have so this is a schematic of the cell membrane, and we have antibodies that bind to the proteins that are present on the cell membranes, and then continue to bind to another antibody, which then binds to the hydrogel matrix itself. And that itself is then connected to a fluorophore. And so the idea there is that, so as we're sort of stripping away the protein, we have the sort of remnant of where the cell membrane protein was. So why is this important? Because this allowed us to capture the regions for where we have cell membrane present. And this allowed us to perform cell segmentation with a high degree of confidence. So we were able to use a toolkit elastic to be able to segment our cells. So from that, what we have is our imaging data. We were able to identify molecules based on the barcodes. And then for those, we also have a coordinate system. And additionally, we have segmentation for given cells. So from that, we're able to calculate for each observed cell, we've got a gene expression vector. So we've got that information, but recall we also have our large time scale time course experiment using single cell RNA-seq. So the question that we first wanted to do was to identify for each of these identified cells, could we perform a classification of the cell types themselves given our curated list of our sort of highly curated vocabulary, if you will, of our cell types. So what I'm showing here is, that UMAP from before, from our single-cell RNA-seq, as well as this sort of UMAP of our seq-fish data using this subset of genes. And what we did there was perform an integration analysis. So this was like a horizontal integration where we restricted both data sets to the set of overlapping genes, and we performed integration using mutual nearest neighbors. So now what we see is a UMAP representation of our joint data sets together. And what we can observe quite nicely, I guess, is that we see a high degree of overlap between these sets of cells. And we would expect that because these come from the same, the same sort of time points, and they're all wild type. So what does this allow us to do? It meant that we were able to classify the cell types of our spatial cells just by using classical machine learning. So we did one of the simplest and most effective techniques, KNN. And from that, we have our originally labeled data, but then we were able to transfer the labels of our seq-fish cells. So that then allows us overall to take our spatially resolved data, and using the same vocabulary, we're able to essentially paint on the cell types for our cells. And what we found overall was that it's sort of highly consistent with what we would expect given our known biology. So, we're seeing like the green area corresponding to the forebrain, midbrain, hindbrain, kind of where the head should be. So that's good. And, the cardiomyocytes around the heart area, we're seeing the endoderm, like the gut tube that's about to develop, and as well as what's going to develop into the spinal cord and all of that system. So that's nice, and it meant that we were able to perform sort of downstream analyses with the same set of our sort of same understanding as before. So I want to highlight just one sort of key bit from this analysis. So yes, what can we learn from this data integration? So what we performed firstly with our spatial data was to try to understand how much transcriptional heterogeneity could be explained by spatial location. So here what I have is a set of violin plots of genes grouped by the cell types. And on the y-axis is the t statistic corresponding to a linear model where we use the a given cell's gene expression as the response and its neighboring cells gene expression as the predictor. So if we have a high t statistic, that means that we can very well predict what is that cell's gene expression. And so what we found overall was that we had a bunch of genes that were quite high, sort of highly predicting of these neighboring cells within that brain subcluster. And so then that sort of hinted towards us that actually this subset of cells may be much more heterogeneous than we thought. And so from that, we looked at this group, and what we were able to find was that we could subset further into groups corresponding to the forebrain, the midbrain, and the hindbrain. So that was quite helpful for us because it meant that we were able to perform this expression-based clustering, but then actually check and see where anatomically it corresponds to, and that itself is not so clear if you only have the dissociated single-cell RNA-seq data. So So that itself was helpful, and this is our embryo 1. What we noticed in our second replicate was that we had quite a nice capture of this region called the midbrain hindbrain boundary. So if I'm sort of showing here, this is our second embryo, and if I zoom in, we have a mixture of cells corresponding to the midbrain and the hindbrain. And the key question that sort of arises from here is, what level of changes in gene expression do we see across space, right? So along here. So what we did was take each of the cells corresponding to this area. So now we have these given cells corresponds to this midbrain hindbrain boundary area, and we performed a diffusion embedding here. So along this diffusion component 1, we're capturing how much sort of smooth change do we see across space. And then what we can do is extend that out back into our spatial coordinates. So what we're seeing here is the cells are now colored by the diffusion coordinate 1. So you can see along here that cell type, that cell color is matched. Oh, it's my necklace. Let's see. That's what's happening. Back on?
Yeah.
Okay, so apologies, the offending necklace has been removed. So thank you for putting up with that rustling. I hope that's a little bit more smooth for the rest of the talk. So what I was saying was that here we've got our cells colored by the diffusion component 1. And here I've overlaid a vector field. And so this is not RNA velocity, so keep that in mind. This is simply a vector field where we have the direction corresponding to the biggest change in the diffusion component over space. Now, if we had very sort of stochastic, highly variable, not correlated with space expression, we would be expecting to see something like a speckled pattern. But instead, what we see is this very smooth change, and very much concentrated around this boundary area there. So that itself was quite interesting. And one of the follow-up questions from that was, which genes are actually contributing to these changes in this space? And I mean, of course, we've got different axes as well. So, we have the dorsal to ventral axis, but we also have the sort of floor plate to roof plate axis as well. So what we did there was to use a technique called SC-HOT. So this is a method that I developed for performing higher order testing. It's on Bioconductor now. It's got a hex sticker as well. That essentially tests for changes in variation or covariation in space. And so for this, I used the weighted mean as the input here. So here we're now looking at which genes do we see there being a high amount of changes in space in terms of the weighted mean in any way, in a non-parametric way. And so here I've got a plot of the mean expression by the shot test statistic. And here I'm showing the spatial plots of gene expression for given genes. And note that from this, we were able to perform a gene imputation as well using our single-cell RNA-seq reference data. And I hope that we can sort of appreciate that there's quite a range in terms of the spatial patterns of expression that we're seeing. So it's not just one type of, emanating out from the MHB. We've got specific patterns across some areas and so on. So, we can see quite a bit of stuff happening there. So that was quite of interest, and it's something that we can learn a little bit more from our data, after having understood it with our single cell RNA-seq in mind. So from this, the spatial mouse atlas is a resource that can be examined. I created a interactive expression browser for this, and it's available to look at over here. So, in terms of visualization and interaction with these spatial data sets, I did find that there were sort of three components that were quite of interest. First is to be able to look at expression in space. The other is to look at the individual molecules as well. So recognizing that, of course, we have different scales of our original data, not just the cell totals, but also the molecules themselves. And then in addition is, this idea of, how do we actually subset our spatial data in meaningful ways? So, from this, we could perform a virtual dissection and then on the fly, differential expression analysis. So, this is an example of an expression browser, and I'm certainly seeing many more sort of wonderful expression browsers come online. So, if you want to have a look at what else is possible in the realm, please, have a look at Federico's great list of browsers. So, here I've spoken about, we've generated a spatial data set, and we used our single-cell RNA-seq to help us understand our spatial data. But what if I flip that around? So, as we generate more spatial data, what can we learn from these spatial references? So, what we're essentially trying to do is, go around the other way. So as we become more and more as we have these, then if we have, say, new dissociated data that we don't necessarily know that much about, how could we learn? And so this leads to one project that was spearheaded by Elias Haydari. So he was a master's student who visited Cambridge and is now doing his PhD here in Heidelberg as well. So Elias developed a technique for spatial reconstruction, and this method is called SageNet. And so how does it work? The idea here is that, we have sets of spatial reference data and putting two things together. So first is estimated gene interaction network, as well as a non-overlapping partitioning of our spatial data. So putting it into tiles, into different partitions, means that we can then train graph neural networks to be able to predict for any new gene expression vector where it best corresponds to in terms of the partitions themselves. So that ends up with a trained model, as well as a way of being able to interpret the model itself. So from that, we can understand a little bit about spatially informative genes. So yes, So then how do we perform the spatial reconstruction of, say, dissociated data? So if we say started with a query, then what we would extract is our predictions based on the various partitions, which can then be transformed into a predicted cell-cell spatial distance matrix. So we're sort of changing from the expression space similarities into a predicted proximity of given cells. Now, what Elias also did was enable the use of multiple spatial references. So, you know, you don't only have one particular picture, you know, you may have multiple. And, you know, for our spatial reference of our embryos, you know, we have 3. And so, you know, it could be that we've got 3 particular references that we may want to use. And so there we perform a model ensemble to be able to get, in the end, an aggregated cell-cell distance matrix, as well as associated confidence for a given cell. You know, how well do we actually know where it could land in terms of the latent physical space. So to illustrate this, one sort of component here is, you know, if we go back to our sort of organogenesis data set, you know, this is a T-SNE representation, very similar to the UMAP of our cells. We can see our green region corresponding to the brain subtypes, and these colors here correspond to the data set. So we have our single cell RNA-seq as well as the different spatial references. So that's normally just using gene expression. But if we use SageNet using our ensemble from all 3 of our replicates as our reference data sets, then what we can see is that the sort of recapitulated, you know, our predicted sort of physical space allows us to really distinguish between these sub regions of the forebrain, midbrain, hindbrain cluster. And yeah, so that's one key thing. There's another sort of few things to really sort of look at as well. So I encourage you to look at our preprint. But yeah, so, you know, what we, to sort of, I guess, emphasize that a bit more is like, we're not just getting these 3 groups out of nowhere. These do correspond to the 3 subgroups that we see. So I mean, to me, I think that's a nice example of sort of going back and saying, okay, well, you know, as we're becoming sort of rich in terms of the data sets that have spatial technologies, this is a way that we can go forward with using our dissociated data as queries. So then I want to sort of highlight a bit about, you know, what's happening with integrative analyses as we change with technologies. So I'd say over the last sort of 4 or 5 years, I've been observing 2 key things. The first is an increase in the number of single cell technologies. So here we're seeing a plot of time and then the number of single cell transcriptomic studies that are emerging. But not only that, we're also seeing an increase in the variety of different single cell technologies as well. So this comes onto this other side here, right? So here's a little cell, and you know we're capturing many different types of features, I guess. So, you know, we're capturing things related to the DNA, the RNA, the protein, the lineage, the space, and so on. And so from a methodological sort of computational perspective, you know, if we want to be able to integrate these data, you know, as we add a data set coming from another technology, you know, the set of features that may sort of correspond to that will, become smaller and smaller, and it may be vanishingly small. So this represents a real sort of problem in terms of an integration challenge. And in particular, this is, what we term as a mosaic data integration problem. And so where did this wording come from? It came from this really great review sort of perspective piece about challenges in data integration. So the things to distinguish are, if we look at particular sets of cells corresponding to, say, data sets. Here we've got colors corresponding to whether or not we've identified, whether or not we've measured these features explicitly, as well as the various sets of features, and they could correspond to different assays. So typically in the previous example that I showed you, where we had single-cell RNA-seq and our seq-fish data, I said that we restricted down to those intersecting genes. So what I had done was basically a horizontal integration. So I had pretended that, we only have those features, we don't have any others. But you can see that that's annoying for one, because you think that, okay, well, I could have used these extra features in some way. Wouldn't it be nice if I could make use of that in a meaningful way? And so one way that I sort of hope to contribute to this problem of mosaic data integration is with the development of StabMap. So this is a method that performs sort of stabilized mosaic single-cell data integration by taking note of what are unshared features. So how does it work? So first, we identify a mosaic data topology, we build reference embeddings for given references, and then we perform projections of non-reference cells by traversing this topology. So if I give a bit of an example, a bit of a schematic, I guess. So, say we have our observed data matrices. Along here, we have our cells, and then on the rows, we've got the features. So for this data set, we can see that there's actually no overlap between the first and the third data set. And this sort of middle data set acts kind of like a bridge between these two. So from that, what we can do is infer or extract out a network structure that sort of summarizes the relationships between the various data sets. And so what we have is that there's, an edge between these two data sets because they share features and so on, but then there's no shared features between the top two. And our goal there is to estimate a joint lower dimensional embedding where we've captured all of the cells in some kind of combined coordinate space. So how do we do that? So we first select a given reference data set, and then for a given query, we perform a linear model fitting, where our predicted values correspond to the low dimensional space of the reference set. So this can either be like the principal coordinate scores, or it could be the linear discriminants if we have classes. And then from that, we have our, so the scores themselves are our response, and then the predictors are the shared feature space between them. So that's fine. But then what happens, with our third data set? So then from here, we're performing an iterative procedure of building a model and then training it on our intermediate data set, and then being able to sort of project further onto our sort of target low dimensional space. So overall, that gives us this, in the end, a StabMap embedding for a single reference data set. And if we, say, select multiple reference data sets, then we can sort of use these groups and combine them by reweighting the relative contributions to then sort of achieve our overall embedding of cells. So that's how the method works, and it's currently available as a preprint and on GitHub at the moment. And I want to just highlight a little case study for how we use this method. So I did nothing. So here, what we did was we looked at our sort of mouse organogenesis data set, but we had an additional data set where we had performed perturbation of a particular gene. So here we have cells that correspond to a knockout of the gene brachyury, and these ES cells were injected into a mouse blastocyst, and that develops into a chimera embryo. Then from that, this sort of chimera embryo, it has a mixture of cells that are wild type for this gene and mutant for this gene. Then we can perform single-cell RNA-seq and be able to, look at the gene expression profiles as well as identify which it came from. So you can see that this corresponds to a mosaic data problem because as we look at our sort of data set, what we have is, the set of genes that were not captured in our spatial atlas, as well as the ability to, extract maybe new features. So here, what we did was generate new features that correspond to the average expression of the nearest cells in space. And so, this is, if we had the spatial locations of our dissociated cells, we would have been able to measure that. So these are sort of latent features, if you will. So if we run StatMap and we look at our data, so here I'm showing a UMAP of our cells, and these cells correspond to both the single-cell RNA-seq as well as our spatial atlas. What we sort of nicely see is that there's a decent amount of overlap over these two. We would hope that would be the case, as well as being able to distinguish between different cell types as well. And we're seeing some some sort of spreading of given cell types. So it's not purely just gene expression space, it's also taking a little bit of that sort of spatial into account. So from that's nice, but we had perturbation information from our single-cell RNA-seq. And so what that meant was that, using this embedding, we could perform a local enrichment analysis. So what we're seeing here is the same UMAP, but each of these cells are corresponding to the relative enrichment or depletion of the wild type or knockout cells within that embedding. Right, so blue here corresponds to there being not so many knockout cells as we would expect by chance, and then red corresponding to an overabundance than we would expect. So what we noticed sort of interestingly was this subset corresponding to the splanchnic mesoderm. And the reason why was that this actual group was kind of bimodal in terms of the breakdown of enrichment coefficients that we observed. And so if I plot these in space, and so note that, I have the enrichment values corresponding to our single-cell RNA-seq, but thanks to the sort of integrative component here, I can predict what are the corresponding enrichment coefficients in space. What we can see is that we're seeing a shift of the enrichment from overabundance to depletion along the anterior to posterior axis of the cells in the splanchnic mesoderm. So this is the cell type that's going to develop into the tissues that line, your vital organs. And so I think that this is, quite nice because it gives us a way of performing like a spatial enrichment, having taken a perturbation information from dissociated data. So we didn't necessarily need to perform a spatial perturbation experiment, right? And so this gives us a little bit of a hint, and we can, follow up on this and then go and examine a bit more. So I hope it's neat to me, at least. So I guess sort of to close up a bit, in terms of, facilitating these integrative analyses of spatial genomics data, I introduced sort of 3 methods that, are very much inspired by, the scientific questions of, how can we test this? How can we predict this? How can we integrate this? But, the key thing is that to actually be able to perform these analyses and be able to perform, meaningful comparisons with, say, other methods or other data sets, we're using the infrastructures that have really come out from Bioconductor. So things like Experiment Hub, the Spatial Experiment Container, MultiAssayExperiment. And I do wonder if there's scope potentially for something like a mosaic experiment kind of idea. So maybe we'll think about that a little bit. So to summarize, we examined mouse organogenesis at quite high resolution, and we were able to identify sort of spatially relevant genes using SHOT. We sort of enabled this sort of cycle of learning from a spatial reference onto dissociated data using SageNet, and sort of addressing this issue of mosaic data integration with StatMap. And I think that moving on, the idea is that we want to focus on new ways of learning from combined data sources. So I will take this opportunity to plug a little bit. So as Charlotte mentioned, I opened my lab 4 months ago. Over in the University of Sydney. So I will say that I'm recruiting. At some point, postdoc, and there is a PhD position open at the moment. And so with that, I'd like to thank the wonderful group of people that have been involved in the number of projects that I presented today. And thank you once again to the organizers, and thank you for listening.
Thanks a lot for this great talk. We'll take some questions from the audience. I just wanted to say for the people who may be following online, if you have a question, you can go to the conference website. There is a Slido link and you can post your questions there. So are there questions here? Microphone.
Hello. Thank you very much for this nice presentation. I had a question about StatMap.
Yes.
So you showed a very nice example combining single cell stack with Stackfish, but yeah, both technologies are kind of capturing the same omic, let's say,
yes,
which is transcriptomic.
Yes.
I was wondering, would you advise using StatMap with different omics? And I have in mind, for instance, single cell RNA-seq with proteomics, where the correlation between the two omics is maybe not so one-to-one.
Yeah, that's a great question. Thanks for asking. I think the key thing there is that, so long as you have some shared features. So for example, you said like protein and RNA, you may want to be integrating, say, like a single cell RNA-seq with site-seq data or something along those lines. And so there, your shared features may correspond to the gene expression, right? So the expression of a given gene. And so I would say are sort of like truly shared. So yes, that's certainly possible. But say you had something else, right? Like say you've had like cytoff or like imaging mass cytometry data where you have proteins and you have your single cell RNA-seq. So that, in my mind, it corresponds to a diagonal integration because you don't actually have sort of true shared features, right? How you would be able to transform this into either a horizontal integration or a mosaic data integration is to make an assumption, right? So you need to assume that, you know, the central dogma holds true for all of the genes and all of the proteins. And, you know, sometimes it's good, sometimes it's not. But yeah, that's what I'd say.
Hi. Thanks for the wonderful talk and all the great work that you've done. It shows here. I have a kind of like more general question, I guess. So I have the feeling there's this sort of like, we're on the verge of this like new sort of like revolution with spatial transcriptomics, and especially in the imaging world. And I guess Bioconductor in the last few years has been very focused on sequencing data. And so I wonder what you see as the future of Bioconductor is like R, the role of R with like cell segmentation and these all imaging techniques, and as a consequence, the role of Bioconductor. And the second question is, what is the raw data in some sense? So I was really happy to see that you shared all your mouse data in this nice Shiny Hub.
Yeah.
And this is gene count, or I guess gene count and transcript locations.
Yes.
Do we need the images or not?
Yeah, no, it's an excellent question. So maybe I'll address the last one first. So yes, the image data, like the sort of raw data, is a stack of TIFFs, right? And they're available. I'm in the process of putting them on the image data resource, the IDR. So there, you know, it'll be available as OM and GFF format for each of the tiles. And so then that corresponds to the images as well as the segmentation masks as well. So yes. Now, I mean, the next question is, you know, how do we deal with imaging data in R and in Bioconductor? And I mean, I think that there are, you know, there's previous work here, right? Like EBI image, right, is an example. And there are people in this room who are actively working on this who can answer much more with much more detail. But yeah, like I can certainly agree that it's happening, it's coming, and we as a Bioconductor community are working on it. So yeah, I mean, we're going to see more throughout the next few days, especially with the workshops. You know, for example, the Niels's work with Cytomapper and so on. So yeah.
So I'm also tempted. I think the image data is of course interesting, but at some point the images are converted to numbers, and then it really turns into more like a spatial statistics problem. Yeah. But I mean, so my real question that I wanted to ask you, I was intrigued by your SC hot statistics that many of the really highly expressed genes were very flat. I don't know whether you have the plot still there. Yes.
So what you were referring to was the plot of the x-axis being the mean expression, and then the y-axis being the test statistic corresponding to SC hot. And I guess it's sort of similar to maybe like a kind of like in the same way that you would interpret like an MA plot, right? Like if everything is up, then everything is up together, right? So that's the sort of thing where you're seeing, like, we observe indeed some genes that are just very highly expressed, but they're not actually varying in their mean expression over space. But yeah, it's the ones that have this mix, which could either be spatially associated or it could be not, it could just be random. Yeah, so it makes sense why we're seeing that relationship. Very cool question.
Hi, so thank you for the very nice talk. So I'm going back to the integration part. So I'm reanalyzing a lot of different single cell RNA-seq data sets at the moment, working not with human and mouse data, but with all kind of non-model organism. And for me, even the data integration starts a little bit before. So coming back to the features, the problem most likely is that you will not have for most of these analysis information of the gene annotations and if isoforms have been resolved or not. Yeah. And that is really a big problem, because in the end, if you integrate the features just by the name, a gene is not a gene. You can have isoforms and stuff like this. So I guess even there, we as a single cell RNA-seq community, we would need to work on integrating that at least these mandatory annotations are integrated in the data sets. I just want to know what's your opinion about this stuff.
No, I mean, I certainly think that it's a challenge, and there are works to address it, or at least try to address it. I mean, yeah, so like there's two key issues, right? There's the annotation, so being able to extract the relevant feature from the sort of sequencing, right? So that itself is important. But then the second is the feature matching between different organisms. And so, yeah, like I said earlier, right? Like you're making an assumption when you're matching features. Like, so then you're assuming that the RNA abundance for gene A as it's annotated in species like X is the same as like gene A in the next species. And I'd say for doing like a mosaic integration, you need to have some set of matching, because you need some core set of shared features. But it may be that there's additional features that are not necessarily need to be matched. So that's where, like, I could see an application of StatMap to sort of like the cross-species dataset comparison, where you could like retain those additional features or not have to, you know, maybe like the many-to-one relationships or the many-to-many. So yeah, it's certainly a good question, yeah.
Thank you, Shila, for the great talk. I really enjoy it. I have a question regarding using these shared features. I mean, do you think that this kind of data you have of shared features by hybridization and sequencing can serve us to do data imputation for kind of using this hybridization information as priors and then constructing Bayesian normalization methods for single-cell RNA-seq? It's useful, has sense.
Sorry, let me understand. Your question is like, could that mapping be used to perform additional modeling?
No, I'm referring concretely to the fact that since hybridization is a different technique, you can have more quality data for little features, but that can serve you as a prior to do another kind of normalization.
Yes, absolutely. Yeah, no, that's thank you for clarifying. And so the thing that you can note is that once you perform mosaic integration using StatMap or anything else, now you're in a space where you can actually perform downstream horizontal integration, right? So you could perform, say, like a batch correction on top of that to address any other sort of relationship. So yeah, absolutely. Yeah, there's a question here, but aware of the time.
Yes, just checking if there were any questions online. I think we'll wrap up here. You will be around, right, for the conference?
Yeah,
so
absolutely.
If people have questions, they can just come back to you. So thanks a lot again for this great talk.
Thanks very much.
So the next session starts at 10:30. Don't go too far. I mean, there will be a coffee break after that session, so don't go too far. And that will be a short talk session. So.
Oh, we should write EuroBioC Twitter handle here. Right. Because on the YouTube, you see the this blackboard. You don't actually see that, so it'd be a really good idea to do.
That's me. Yes. Okay.
Andrea here. Welcome to the short talk session today. For those who don't know me, I'm Aideen Culhan. I'm co-chair on the Community Advisory Board. So if you're interested in learning more about the Community Advisory Board, please catch me at coffee or Twitter message or whatever. And so our first talk is a really exciting talk from Andrea Sottosanti in Davide's group. So it's going to talk about co-clustering of spatially resolved transcriptomic data. Thank you.
So good morning, everyone. Thank you to be here. I'm Andrea Sottosanti. This is a joint work with Davide Risso. And with this work, we want to propose a new method to analyze spatial transcriptomic experiments. In particular, does it work? It's not working. It's okay. I go with this one. Okay. In particular, when I talk about spatial transcriptomics, I mean, this is an example of an experiment with a Visium technology. And so we place, the idea is placing this tissue sample over a grid of spots. So this is the chip. And at the end of the game, you have a data matrix of dimension n times p, where n is the number of genes which are processed, and p is the number of spots which collect the signal coming from the genes. So with this kind of data, people started to become interested in the so-called spatially expressed genes, which are genes whose expression in a single spot is somehow influenced by its expression in the surrounding spots. So these are some examples. But at the same time, in a very parallel framework of the literature, people started to develop methods in order to cluster different parts of the image coming from this kind of experiments. But we think that these two parts of the literature should be seen as the same thing. In particular, what we want to propose here is like to solve three possible tasks. The first one is trying to perform a clustering of the image coming from an experiment using not just the expression of the genes, but also using the spatial coordinates, which are provided by this new kind of technology. At the same time, because we induce a clustering of spots, so of the image, we want to perform a clustering of the genes using the information coming from the fact that the image has been clustered. And third, once we divide the image into clusters, so into areas, we want to also analyze which are the highly variable genes in each and every part of the image. So three different tasks, but one single method in order to perform this kind of analysis. We use a co-clustering method, which means that we perform a clustering both of the rows and of the columns. So the idea is that once we perform, this is an example of two clusters of genes and two clusters of image areas, we end up having, after reordering the rows and the columns, we end up having a data matrix divided into blocks, where each block represents a certain group of genes which are expressed in a similar way, just in a specific part of the map. Okay, this is the meaning of a single block. And so now it comes proposal. We call it spatially resolved transcriptomics co-clustering, or Spartaco. The idea is that we perform a double clustering of the rows and of the columns, and we then model the expression of a gene just in a specific area of the map in this way. As you can see here, there is a constant value for the mean, but the interesting part is the variance, because of we specify a gene-specific variance, so it's something just related to the gene, but also we have this component, which is a matrix, and it tries to capture the spatial variability into the data, and also it allows for a possible residual variance, which is called nugget effect. So now let's see an application of this model over this spatial IBD data contained into the spatial IBD package provided by the Library Institute for Brain Development in Baltimore. So we started with almost 34,000 genes measured over more than 6,300 spots. So we reduced the number of genes using this pre-processing method proposed by Tuomas and co-authors. The idea is not just trying to select the most informative genes, but also trying to transform these genes into a continuous measurement in such a way that we can apply our model. So let's see now some results. On the left, you can see here the average expression of just one cluster of genes. In particular, as you can see here, we isolate those genes whose expression is larger in the white matter area, which is this part, and become smaller when you go over the rest of the cortical layers. On the right side, you see the partition of the image into 9 possible clusters. So, and in particular, you see that the white matter area and the border with the rest of the cortical layers is identified by the red dots, the black dots, and the gray dots. But what is interesting is like that right now, because we performed a clustering of so 2 clusters of genes and 9 clusters of the image areas, we now divide both the genes and the image areas. And you can see on the left side that the average values of some genes in some parts of the image is different. But also it's interesting, and I want you to concentrate on this right side image, that also the signal, the spatial signal coming from this data is different. For example, as you can see here, this is a cluster of genes, and which is denoted by k equal to 2. And you can see here that the spatial amount of signal is larger in the clusters, so in the areas of the image denoted by 1, 7, and 8, which indeed are the part within the white matter area, and is smaller outside. So we identified in this way some genes which are highly spatially expressed, but not over the entire tissue sample, but just in specific areas, which is something that was not possible to be done with like the previous methods in literature. And also, once we separate the spatial variability, we can now concentrate on the gene-specific variability, and in particular just exploiting this quantity, which is like the conditional distribution of the gene variance. We can isolate the highly variable genes in each and every possible part of the map that has been identified. For example, within the white matter area, there is signal that these genes, the MBP and PLP1, are highly variable. Or for example, if you move to the cortical layers 3, 5, and 6, they appear, these genes CCK and PCP4, so with these very nice patterns. And also, like with respect to the other methods that are used, for example, to sort the genes in terms of like how much they are informative. So as I told you, we used a pre-selection method in such a way that we could select the first 500 genes. And as you can see here, so the genes are sorted from like the most informative to the less informative. But as you can see here, our method is able to identify some genes which are interesting, so highly variable, even though the pre-selection method placed them at the very end. Okay, so it means that we are just looking to something which is different with respect to the method that are being developed in literature right now, with like specific patterns just in some of the areas of the tissue sample. And so we can end up having a list of the highly variable genes for each and every region that we discovered over the tissue sample. So to conclude, this is like a model that is able to simultaneously cluster the genes and the image area of a spatial experiment, exploiting the mean, the variance, and also the spatial covariance of the data. So we developed also an R package, which is available online. We are still working on this in order to speed up the computation, but it's already available. And so this is a presentation that, like, the content of this presentation comes from this paper, which has been just accepted on Annals of Applied Statistics. So feel free to reach me out or like see my website, and thank you very much for your attention.
Questions? Wolfgang?
There's one question I have here. to what extent do you assume that there's a smooth variation of expression of a space, and how do you deal with edges or your more sharper boundaries?
You mean like? Can you repeat, please?
I mean, to what extent do you assume smoothness of the expression over space versus sharper boundaries?
Okay. Oh, but well, the idea is that like because of so the expression is like taken into account by the fact that we insert into the model this quantity, which is a covariance matrix. So the idea is that like yeah, even though it's smooth, like the amount of information and the spatial variability is captured by this parameter and this covariance matrix. So yeah, I know that at the very borders, it's quite difficult to probably estimate correctly the spatial signal. But I mean, this is a covariance matrix, and so we kind of isolate the spatial information into this quantity. So it's like a parametric model. So I don't feel you really have this kind of effect at the borders. Some problems at the borders, I think.
Thank you. For those online, the Slido code is 1680070. If you want to ask questions, there isn't at the moment.
Can you please repeat, sorry?
Oh, I was just giving the code for people online for Slido. Sorry. It wasn't a trick question. Thank you. Now we have a very interesting talk from Ali Oghabian from Helsinki, talking about retained introns and the package you've developed interest. Thank you.
Okay, so hello everybody. I'm going to talk about splicing analysis using interest, a package that I developed. I would like to first thank the organizers for providing me the opportunity to present my work. Most of the work that I'll be talking here was done in collaboration with Dr. Mikko Frilander during my PhD. I'm currently now working at the University of Helsinki and Folkhalsen Research Institute. I would like to thank Magnus Erdner Foundation for funding my postdoctoral research. So in eukaryotes, genes are constructed of anexonic regions. Transcriptionally or co-transcriptionally, introns are removed, exons are combined to one another, eventually building the mRNAs, which could either code for proteins or have other functions. So alternative splicing is how different combinations of the exons of a gene can make multiple mRNAs for a gene. So we have the cassette exons or exon skipping, alternative 5', 3', mutually exclusive, and the one that I will be mainly focusing today on, intron retention. So intron retention is associated with weak splice sites, small intron sizes, high GC content, several splicing enhancer, and inefficient intron splicing. mRNAs with retained introns are known to be detained in the nuclear and eventually degraded. So usually people do gene expression analysis. So the changes in transcription of a gene. Sorry. So they want to see the changes in the transcription of the genes across several samples. They want to see whether genes are more transcribed or less transcribed. However, the genes may be similarly transcribed. However, they might be spliced differently. On abstract level, I feel like it's like comparing two slices of cheeses. Some holes will be similar, some holes will be different. We're looking for the holes that are different, but the story could be even more complex. Genes may be transcribed differently, and be spliced differently as well. So different software packages exist for carrying out these analyses. Some of these might be familiar to you, the DSEC2 HR for gene expression. In addition to alternative splicing analysis, several packages such as mine has been developed that specifically study intron retention. And you might think to yourself, why would anybody do that? And the reason is that because intron retention analysis is difficult. Introns are lengthy. A lot of reads are required to cover their whole landscape. They cover non-coding RNAs. They have repetitive sequences within them to which small number of reads map, and mRNAs with retained introns are lower expressed, difficult to detect. So I developed interest and is actually available in Bioconductor. It accepts BAM files which are produced during alignment, and it accepts it as inputs, and it provides several tools, several methods in order to summarize read counts that are useful for splicing. You could count for each exon the number of reads that skip the exon. You could count for each intron the number of reads that span the intron or map into the intron. And also it does also provide functions based on the familiar DSEC2 and HR that allows you to do statistical tests and detect those introns that are significantly retained in some samples in comparison to the others. Some visualization tools also do exist on the package. Psi is also measured by the software, so it's actually a percentage spliced in. How it calculates it is based on the reads that map to the intron exon junction, and the reads that span the introns is via this formula. It's just basically the relative of one versus the sum. Twice the NEE actually. However, for the significant analysis, what we do is that we usually also do consider the reads that fully mapped into the introns. In the past, we have used interest for analyzing the retention levels of the U12 type introns. These introns are in about 700 to 800 genes in human only, and they are spliced via minor spliceosome. Here you see two sets of data. So both of them are basically diseases from patients that have diseases that are caused by defective minor spliceosome. In one, defective splicing has led to elevation of the intron retention over all U12 type introns. In the other one, in only a subset of U12 type introns, those U12 type introns with weaker splice sites. However, in the latest version of the interest, we have basically also provided capability to measure psi, but exon inclusion level. So this could be useful for other splicing analysis than intron retention. And one thing to keep in mind is that sometimes these defects are in very few samples, like we're studying rare diseases. And also some genes are just so complex, like this TIE10, mutations in it could cause neuromuscular diseases. And it's a huge gene with 366 exons. So it could help that every now and then just plot basically the exon inclusion levels across the whole gene for several samples and try to focus on those areas that have a higher variance of exon inclusion level. Another thing that we're trying to add to the software is basically to make it strand aware. At the moment, it isn't, unfortunately. So if you have a gene that is on strand, it's on sense overlapping another gene, which is on antisense, and if you have reads mapping to both of them, if you know that read is ordered on R1, F2, you could figure out from which RNA it has actually sampled from. In addition to that, I'm also trying to add a wrapper function, which of course is going to be called interesting if you have a function, if you have a package called interest. No. So what it would do is that it would because a lot of features are now being added to it. So for the requested method by the user, it will run the right sequences of the functions, and these could be sometimes quite time consuming. So I'm thinking of strategies that if somewhere here it finalizes and crashes for some reason, the user next time could provide results that have been written out at this point as a kind of parameter, and if it sets those, it would just skip these parts. I'm looking for good suggestions on how to implement this. If you know, I'll be interested. So with this, I would like to thank you all and take your questions if you have any.
Thank you. Any questions? Yes. So yeah, once you find a differentially spliced gene using interest or one of the other methods for alternative splicing, so what can you do downstream of that jury? What would you do with this information to move forward?
So I'm guessing that you're wondering about the application first. Sometimes you will find mutations that we don't know what is the cause. It is nice to check the splicing. If it's somewhere in the introns, you could figure out that actually is causing defects in splicing, which could be the reason of the disease, as for instance. Sometimes you have these, in our cases that I showed here, the two samples, they are effects that the spliceosome itself is defective. And it wouldn't be a complete study if you don't read it completely through and see exactly that the splicing is going wrong and the whole story there is revealed that way. Did I answer your questions? Okay, cheers.
Yeah, thank you for your nice talk. So I would have a question. The critical step I see is, of course, the mapping. Are you providing some tutorials or something like no-goes? What might happen to duplicated genes and stuff like this before entering your interest?
Well, that's the first question. So we're not really dealing with the mapping ourselves. We have some recommendations if you're using STAR, and I think I have provided a couple of recommendations. I think in the documentations it could be improved to provide even more recommendations, but we're basically leaving it to the mapping softwares themselves, because this, if we want to cover the whole thing, is going to just get too enormous on it. But yeah, that's basically how we're dealing with it at the moment. Yeah.
Thanks a million to all the speakers in the session, and congratulations to the speakers for actually staying on time. That's really wonderful, particularly when we have coffee break now. So enjoy the coffee break, and you may ask more questions of speakers then. Thank you.
tinyurl.com/EuroBioC, alles klein, 2020. Ja, genau. 2020, ja.
Soll ich einen Adapter holen?
So we stop the sharing here.
we have enough more than 100. Das ist ganz viel. So, jetzt kannst du den Screen sharen. Hast du Audio frei? Oder nur Video?
Yeah. Test.
Ta-ta. Ta-ta-ta.
missed. See for you, mute. Yeah. And yeah.
Okay, welcome to this session. We are going to have speeches from people from Germany, Finland, and Spain in this session. I'm moderating this. My name is Leo Lahti, and I'm a member of the Community Advisory Board. So our first talk will be by Constantin Ahlmann-Eltze from EMBL Heidelberg from the Huber Group, and he will talk about single cell studies. Thank you.
Yes. Hi. Thank you very much for the nice introduction. I'm Constantin, and I'm from the Huber Group at EMBL, as was said. And I want today to present to you a new framework which we've been working on for analyzing multi-condition single-cell data. And to illustrate this new framework, what I'm going to quickly introduce to you is the data which I've been looking at. So this is some data which has been generated by collaborators of ours who've been looking at the effect of gene perturbations on the Drosophila intestine. And if you just do a UMAP of the raw log transformed data, you see that you have the different perturbations. You have 3 different perturbations and the wild type, and they already cluster mostly together. However, there's this one group of cells which behaves a bit differently. And currently, if you want to analyze such a data set, what would most typically be done is that you start annotating cell types. So here I'm showing the 2 most prevalent cell types in this data set, which are enterocytes and enteroblasts, shown in red and green, and then some other not so interesting cell types. And subsequently, one could be looking for differentially expressed genes. So, for example, detect that lysosome B is differentially expressed under the perturbation in the green enterocytes. Sorry. However, and in the next step, one could also look for more fine-grained structure, for example, plotting the expression values on the UMAP to see if there are structures which don't exactly correspond to the annotated cell clusters. And what I want today to present to you is an alternative approach to do this, which doesn't require any kind of clustering. So where you don't need to annotate the cell types beforehand, but where you do the mapping and differential expression without this. So the idea is that you would integrate your data and in the next steps look for differential expression, but just try to figure out what are these matching cells and how does the expression for example, the lysosome B cell gene change across the cells. The good thing is that if you would follow this approach, you don't need to do any cluster annotation, which can be very tedious because, for example, if you can think of a cancer sample, there might be no good reference atlas available. And although there are already many different methods for doing data integration, I want to present a new method, which I think is arguably simpler and thus allows more flexibility for doing downstream analyses. Because the good thing is if you try to stick without cluster annotation and without cell type annotation is that you can also find differentially expressed genes which don't neatly map to the obvious boundaries of cell type. So we have this cluster up here, the cell population, and we see that this gene amylase D is only differentially expressed in the right half of this cluster. So you could now dive in and try to see, is there something specific about the right and the left half of these cells, and why would this kind of pattern arise? And if you look up in the literature, so this is now I mentioned in the Drosophila midgut, there has already been examples been described where the knockdown of or knockout of the X-box binding protein 1 has been associated with this change of the amylase D expression levels. So how do we go about finding this? I have made this small animation, so let's hope this works. No, this doesn't not work. So sorry. I am drawing dots instead of having this. Sorry about that. Does anybody know how I stop drawing dots and get this to play? Okay, so now this is rotating. So sorry about that. What you're seeing here is the data. So the mental image should be that we have cells located in the gene space. So I'm now drawing this cells in a 3D gene space, but you would have to imagine this is actually 10,000 dimensional, so very high dimensional. And we have our 3 subpopulations of treated and control cells. And then the idea is that we fit a plane to each of the conditions. So in this case, fitting the plane only to the control cells, which are lightly shaded. And then we also fit the plane to the treated cells, which are, oh, sorry, this is going to be a bit tedious. I'm very sorry about that. So we fit the plane also to the cells, so the treated cells, which are outside this plane of the control cells. And then we try to find out what is the best way to rotate these 2 planes on top of each other to get one integrated view of the data where both the control cells and the treated cells are landing on the same spot. Because based on this, we get this neat integration, which is arguably quite simple. So we just have the PCAs for each condition and rotate them most in the direct way on top of each other, which allows us, for example, to get the Drosophila data, the cells which are landing down here, to land within the cell cluster. And now to a bit of the more mathy side. So how do we actually achieve this? We assume that each of these planes can be described as points on the Grassmann manifold. So we have the plane, and the Grassmann manifold is just a mathematical object which is describing orthogonal matrices. And these can be understood as k-dimensional planes. And then we look for an element of the tangent space of the Grassmann manifold, which is actually the rotation. How much do we need to rotate the plane P1 to go to plane 2? And other way around, we can say that P2 is the rotated version of P1 by rotating around vector V. So in the differential geometry lingo, people will talk about the logarithm as an alternative word for finding the direction, and the exponential for the rotation, but this is nothing to do with the familiar logarithm or exponential. This is just their specific words for these 2 concepts. The good thing is now that we have this framework, we can take our standard linear model framework, where we have here on the left-hand side the observed expression values, and we have our observed covariates, which, for example, could be 0 or 1 for treated or untreated, or could be a continuous covariate. And we have our coefficients here marked in red, because these are the ones that we need to fit. And we're going to extend this framework. So we have this exp function, which just means we rotate our plane P by the rotation vector V, which we need to find, which is parameterized also by the covariate xi. And then we have the position in the shared space, so the position on the plane, which we call Zi, which is telling us which cells land close to each other. Cells with a similar Zi, we assume might be of the same cell population. And it's also pretty straightforward to extend this model to a multivariate setting so that we can actually handle not just individual treated, untreated, but much more complex experimental designs where we can account for any kind of batch effects, continuous covariates, by just having it all integrated into this one expression. And now we can also do, based on this, fairly easily the differential expression, which I showed you earlier, where we, for example, look at the change of this gene of amylase D. So we just, for each cell in the position Zi, we calculate its expression value. So we understand this as a function of the covariate X and the latent position Z. And then to calculate the difference, we just take the difference between the covariates if we assume that it's a wild type observation versus a perturbed observation at the same Zi location. And that gives us the difference at position I to calculate how much does this specific gene change. There's currently an R package under development, which has a fairly simple interface. So you just provide a single cell experiment, you give it the design formula where you can specify any kind of linear model kind of design matrix that you would want to have. Currently, there's some function where you estimate the variance based around a bootstrap, where I just resample the observations to get an idea how much the results change. And then you just can do any kind of contrast to figure out what kind of genes are differentially expressed. And with this, I want to acknowledge my group and want to also acknowledge the people who have generated the data from the Decode project, and want to thank you all for your attention.
Thanks very much, Constantin. I think we have time for one question.
My question is very naive. I mean, the method, it's really good. I will try to explore it. My question is about what's the reason behind using log normalizes data instead of other normalization methods?
It's a very good question. And I've worked on this question before, like what is actually the best way to go about trying to like normalize or variance stabilize your data. I think there are many different methods out there. SC transform is a popular method in the server framework. There's a log transformation, which is arguably quite simple. In a previous study, what we have found is that the log normalization performs as good or better than any of the much more complicated alternatives. If you think about the tools which fit very difficult kind of Bayesian kind of models, in our experience, they just don't work as well to uncover the latent structure of the data sets.
Yeah. So I have a question regarding the, how do you distinguish, or like, what are the assumptions, I guess, for when you have two perturbations, like a perturbation, non-perturbation, and the cell types slightly change? Like, how are you able to map those cell types together? What are your underlying assumptions for this?
Excellent question. The assumption basically in a mathematical sense boils down to that we assume that the volume of the cells, if you think about them in the gene space, doesn't change, so that there is this rotation and translation relationship. The idea basically is that the method tries to find the best way of trying to account for this. But if there's additional heterogeneity, so for example, that there's a new cell arising, or that like something splits up due to the perturbation, this would not be mapped on top of each other. So this is a fairly mild way of doing data integration. So there you would just see in your new space that there would just be two separate kind of groups. So it's not as much as other integration methods forcing things on top of each other, but it's a lot more mild because it has really only this assumption that you can have this set volume or area.
Thank you very much. We can continue on this in the coffee break, and I welcome the next speaker, who is Tuomas Borman from University of Turku. He's starting his graduate studies now and has been working on the summarized experiment ecosystem for microbiome data. So Tuomas, welcome, and the floor is yours.
Okay. Whoa, it's decrease volume. Okay, guten Tag. It's very nice to be here. And okay, decrease volume. Okay, it's very nice to be here. And yeah, for those of you that are doing something with the clinical data or microbiome data or multiomics data, well, it's like a Christmas for you. I'm not the Santa Claus, but I'm Tuomas from Finland. And this is clinical microbiome data science with MultiAssayExperiment. So I'm here to talk about this appendicitis case study, then this µSTASIS framework, and as a little dessert or something, we have results from that study. So that study is like a horse that carries us through this framework, I guess. Okay, so appendicitis, it is an inflammation of appendix. Now we know that it can be divided into two different types: complicated, which requires appendectomy, meaning removal of appendix, and uncomplicated, which can be treated with antibiotics. And as we know today, microbes are much more than the cherry on top of the cake. They are like the strawberry jam inside the cake. So it wasn't a big surprise that now we want to know if the microbes explain the type of appendicitis. So this microbiology appendicitis acuta trial, they collected samples, blood samples, and they measured cytokines from them. And subgroup, they also collected microbial samples from removed appendices. So now they had microbial samples and cytokine samples, and then they called us. So what can we do with this data? First of all, we have to get it in a sensible format, meaning that we have to get it in a standardized data container, which is optimized the data container, in which case it depends on the type of the data. For example, clinical data needs a patient information slot, for example. So here we have some data. Does this work? I don't know. Let's not use that. So we have an abundance table there, and then we have a concentration table there, and then patient data. And here we can see that we have also this kind of phylogenetic tree. So for microbiome data, we can create this kind of object, three-summarized experiment data container. So it is like a summarized experiment, but it has also this kind of row tree slot. So this is quite ideal for microbiome data. And as a normal summarized experiment, it also has assay slot and row data and so on. So this is extension to the summarized experiment. So now we have a microbiome data there. Then we can create a cytokine data object. So similarly, we can create a three-summarized experiment object, but because we don't have a tree for that, it is not there. But it is a similar object. So now we have two individual objects. So how can we link them together? The answer for that is MultiAssayExperiment. So with MultiAssayExperiment, we can link together multiple experiments. And we can link, for example, patients together between different experiments. So our contribution to Bioconductor is µAwares, and it is a framework for microbiome analytics, and it is based on a three-summarized experiment class. And because of that, it is integrated to summarized experiment ecosystem. And that means, for example, that we can utilize those methods from single-cell analytics, for instance. So three-summarized experiment, it extends the single-cell experiment object. So it has these slots for row trees and column trees. And this is an ideal way to store hierarchical data. For example, in this kind of microbiome data, we have this kind of phylogenetic trees, but also we can have hierarchical samples, for instance. And if you have used phylosec, it is actually a subset of three-summarized experiment. In addition to that, µAwares methods, they extend available summarized experiment toolkits. So there's different methods, for example, transformation methods, diversity methods, subsetting, and also multi-assay methods for multi-assay analysis, for example, cross-correlation, and much more. In detail, µAwares consists of multiple packages. There's µA, then there's µAviz and µAtime. And also it has a tutorial book called Orchestrating Microbiome Analysis. And it is similar to the OSCA, which is for the single cell analysis. And as I promised, we go through these results a little bit. So indeed, we found some differences. There is a cytokine and microbial differences between this uncomplicated and complicated type of appendicitis. And there's certain microbes correlate with certain cytokines. And also the type can be predicted based on cytokines. And yeah, all these analyses were done by following the OMA book, so you should check it out. So summary, MultiAssayExperiment is suitable for multi-omics. Then µSTASIS extends the summarized experiment ecosystem, and appendicitis can be divided into 2 different types. I'd like to thank the MAPPAC team and µSTASIS team, and natürlich danke schön für Ihre Zeit. Yeah, thanks.
Thank you very much, Tuomas. We have time for one quick question. Okay, maybe I ask one. How would you see the developments compared to the single cell experiment? Like, what tools have been added on top of that ecosystem?
Well, there's those back. So there's these basic tools for transformation, for example, but there's also tools for analyzing the phylogenetic tree, for example. So that's one. Those are 2 things that are extensions.
Okay, thank you very much. We can continue in the break. So our next talk is also about microbiome studies, this time on longitudinal analysis of microbiota stability. And Pedro Sánchez-Sánchez is a statistical bioinformatician from Barcelona. So very much welcome, and looking forward to the presentation.
Okay, I will start by presenting myself. I am Pedro Sánchez, and I'm currently working at IRB Barcelona. Although the work I'm presenting here is not related with the things I am doing nowadays. This work is part from my past internship at Benitez Páez Lab. And I'm going to talk about µSTASIS for the assessment of human microbiota stability across longitudinal samples. As I told you, this work was guided by Alfonso Benitez, but also Francisco Santonja helped me a lot. And I wanted to say thank you to part of you, or to all of you, mainly because of the acceptance of this talk, and also for accepting me for being part of the first new developers Bioconductor mentorship program by Bioconductor. So thank you for it. Firstly, let me start talking about microbiome research. Microbiome research, you know, has continually increased a lot. Thank you to the implementation throughout the years to gain more details in taxonomy and functional surveys. And this can be represented by QTNets, microbiome-based therapeutics such as fecal microbiota transplantation, prebiotics, engineered symbiotic bacteria, etc. Microbiome data captured as a snapshot over time has let us know some things about microbiota. One is the individual specific human microbiota, or in other words, the inter-individual variability. And also another thing is the within subject variability, or in other terms, the intra-individual variability. And we are kind of knowing right now that changes in this microbiota composition emerge as predictors of clinical outcomes or disease forecast. So I have to talk about longitudinal microbiome data, that these are abundance data coming from sequencing over multiple time points. The main characteristic are the high dimensionality, the sparsity or zero inflation, the over-dispersion, the compositionality, and the complexity. And let me firstly talk about compositionality, because Glork and colleagues back in 2016 talk about the compositional way to treat the microbiome data. Concretely, they put out a paper called Microbiome Data Are Compositional, and this is not optional. Well, I'm not that sure if I could say that, but I'm right now kind of letting you know why we thought that it was a useful way to deal with this kind of data for our goal. And concretely, there are unequal sequencing reads that are an artifact of an arbitrary total imposed by the sequencing protocol. Also, the experiments are biased towards detecting one taxa rather than others. So at the end, we find that there is a lack of independence of counts. Also, there are uneven library sizes across individuals and time points. So this data exists in a simplex. Concretely, the Atkinson simplex you can see there. Also, let me talk about temporal variation, because it's another property, the complexity it presents, since there are a lot of dynamic interaction with the host and with environment. So we think that microbiome data, it's not only about getting more data, more time point, it's also about dealing with them in the correct way and with the correct methods. And here I just want to point out a sentence by a pretty nice recent review that methods for longitudinal microbiome data are still at their infancy. So let's keep developing things. And the main goal of this project was to assess intrapersonally short-term to long-term changes in microbial communities, or in other words, to assess the temporal stability. And for that, we created µSTASIS. And µSTASIS, to keep it simple, liberates on iterative partitioned clusters across pirate across pirate samples for multiple individuals. In other words, we perform Hartigan-Cummings algorithm as many times as possible to get the frequency in which pirate samples from the same individual remains together or not. In other words, in the same cluster. However, since I was like moving the packets from CRAN to Bioconductor thanks to the mentorship program, I realized that there was a theoretical gap that I wanted to fill. And it's this case where you have the square in different clusters. I mean, the two squares are coming from the same patients, but in different time points, right? So these pirate samples are in different clusters, but you can see how the distance between them is smaller, than the ones to the respective centroids there in the blue. So I also wanted to put that in the main algorithm for assessing the temporal stability. And just to talk about some results, we applied our tool for assessing the microbiota stability of the longitudinal publicly available datasets. And here you can see in the left heat map how some patients presents more instability in the first time, in the first pirate times. And this is due to this data coming from intensive care units patients, where most of them received the most aggressive antibiotic treatment in the first day, as it has sense. Also, in the right heat map, you can see how the mean absolute error is, I would say, pretty low in general. And this is by cross-validation that the package allows to do it in a leave one out way or in a scaffold way. Also, we wanted to extend our analysis to other data, and here you can see a pretty long data set where they are not patients, but I mean normal human beings having normal diets, but you can see how the stability is normally pretty high, but there are some events for some patients where there is a loss or drop of stability. And here we also saw how a high fiber diet is much or tends to be more related with high stability rather than high fermented food diet. So here you can scan the paper we published and also the repository of the package, and just me led to conclude saying that we studied temporal stability of compositions by iterative clustering for getting a metric that is contextualized, intuitive, and validated. So since tomorrow is my birthday, let me talk by 15 seconds more to say thank you to Alfonso and Francisco for guiding me through this project. Also to Dario Sterbenak and David Rizzo for mentoring me during the program, and also to many other people that are not completely related to this project, but they make me how I am professionally right now. Antonio Pineda, my first mentor in academia, some friends like Pepe, Carolina, Jose, and Leandro. And also thanks to my current lab, where I'm having a lot of fun at Fraticelli's lab, like doing cool research. The IRB Barcelona Institute has a great community, and I have the luck to have many, many friends. So thank you to you all.
Thank you very much, Pedro. Do we have a question? I would like to ask maybe, is your package supporting some of these microbiome specific data containers in Bioconductor like 5 or the 3 summarized experiment?
Yeah, for sure. Initially, the package was developed and submitted to grant, so it only worked with a matrix, but due to the mentorship, I changed it to make it interoperability both for matrices object, but also with the 3 summarized experiment that I think it's the way to go in microbiome data analysis.
Okay, sounds very good. Thank you. Thank you very much, Pedro. And now our next talk is by Xavier Escriba. There is a small inconsistency in the program. His supervisor is mentioned as the name in the program, but Xavier is giving the talk. He's also from Barcelona, bioinformatician, and talking about privacy-aware federated analysis of omics data. Thank you.
Well, hello everyone. Thank you for being here. My name is Xavier, and today I will talk about OmicShield, which is basically a tool to perform federated omic data analysis in multi-center studies. And to do that, we use Bioconductor packages and DataShield. So we are all on the same page. First, I will talk about what is DataShield, because I guess many of you don't know. Basically, DataShield is an infrastructure to perform federated non-disclosive analysis. Federated meaning that we have different centers, cohorts, or whatever you want to call them, and non-disclosive meaning that we cannot obtain information from the individuals in the data sets that we are studying. Okay, so this configuration or this architecture can be used in a multi-center configuration or single center, and it's quite easy. We are the researchers or analysts in this picture, and we have a package in our computer. And we are communicating with the different Opal servers. I will talk about Opal in a minute. And basically, as researchers, we are trying to analyze the data in the servers, but only by obtaining some statistics. And the important thing is that the data is never on the researcher computer, so it's always on the servers. We have many active users of the infrastructure, as you can see, many projects, and also some pilots. And regarding Opal, it's basically the DataShield data warehouse, we could say. It's where we have all of our data and an R server, which is performing all of our computations, which is kind of handy. Also, of course, for the legal teams that we all have in our research centers, there's plenty of security implemented, some sort of logging for auditing purposes and whatnot. So now that we are a little bit on the same page, let's talk about what I have implemented and what are the new functionalities for DataShield. Basically, here's the list. We could say that we have focused more on the genomic part of things, you see, with GWAS, polygenic risk scores, and so on. but we also have some implementation for differential gene analysis, basically using Lima and Boom. And of course, we bundled a lot of visualization and some sort of pipelines to help the researchers. And to do that, I had to program or to incorporate inside the architecture new data formats, of course, for the genomic data and counting data and RNA-seq. Basically, so far we have support for BCF files, expression sets, randomized experiment, which are basically the standard, I guess, for this kind of data. So how do we do this? Of course, I'm using Bioconductor packages that by design are, of course, not designed to be not disclosive. So we had to solve that in some way. My idea was to basically wrap all the functions with some sort of filters. Basically, we have 3 types of filters: disclosure traps, some sort of filtering, and finally, my last work has been to develop and implement differential privacy inside Omic Shield, which, if you don't know about, it can be summarized as adding control noise to the statistics we are getting. In that way, we cannot obtain whether an individual is present on the study dataset or not. And by doing that and putting all this together with the Bioconductor functions, we are creating a little bit of a trade-off. Basically, of course, as a pro, which is the main goal, is to reduce the risk of information disclosure. Of course, risk zero does never exist. That's quite important to know. And the good thing is that the parameters that control the risk disclosure can be configured by each of the cohort. So for example, if we are in a study with people from Spain and Portugal, and they have different views on the risk they want to assume, they can configure it differently and still use this data in parallel and get a good study. And as a cons, of course, we are adding a little bit of overhead computations, especially in the case of differential privacy. It's a little bit more slow than running the function in your computer like you used to. And sometimes if you are trying to minimize risk, you are having a trade-off because you are losing statistical power. So it's important to have a good understanding of that and find a good concept between all the consortia, basically. To visualize this a little bit better, I have created a very simple flowchart. Let's say that we are a researcher, we are in Barcelona, and we want to use the data from a hospital in Porto that has its Opal server with its data. You can see data is on the server and never on the researcher computer. Basically, first we have this kind of prefilters that I've called. If everything is okay, we go to the Bioconductor function, let's say performing a GWAS, getting the allele frequencies, whatever. And if everything is okay, we do the post filters, differential privacy. Maybe we have to remove something from the data that we're obtaining. It depends on the operation, of course. And if everything is good, we just return the non-disclosive results to the client. Having said that, I will present a very small use case, just GWAS, easy enough. I have taken data from the Cineca project, genotype data, 800K SNPs, and I have divided into 3 different cohorts with these patients in each cohort. You can see like the graphic describing the configuration is pretty much the same as I showed you before. So of course, when you do a GWAS, you obtain the results and you want to visualize them a little bit. I have bundled a typical Manhattan plot in the function. And of course, now we have the result for the 3 cohorts, but there's more to do when you are doing this kind of analysis, as probably many of you know. For example, we included the method suggested by Winkler et al. In a paper by Nature Protocols, in which he proposed to have different plots in which you can see whether your data is good enough or if it's well balanced between the cohorts and whatnot. Basically, I have taken his methods and implemented them in Omic Shield. So you can run the whole pipeline with just one package, which is quite handy. For example, we have tools to see strand issues with straight transformations, reveal issues with beta estimates and whatnot. All of this is all together, so that way you only need omics shield to perform the pipeline. Then, of course, we wanted to include a little bit of meta-analysis. I thought it was useful since we are dealing with data from multiple cohorts. I chose to add beta coefficients and Fisher's method. But the good thing is that since you are getting the results, if you have your own pipeline with metaphor package or whatever you like, you still can use it. For example, I just plotted the results with the Fisher's method. And as a little bit of summary, basically 3 take-home points. With omics shield, we are trying to create easy non-disclosive omic analysis. In that way, hopefully we will accelerate omic meta-studies because, as probably you know, the bureaucracy and whatnot is quite slow. And also we are trying to develop curated pipelines which contains the relevant steps, such as meta quality control, for example. And if you're interested on the future of the project, basically, I will try to explore a little bit newer or more state-of-the-art techniques for non-disclosive and work a little bit more on RNA-seq and single-cell data, so it all can be introduced. Finally, some acknowledgements to the people that helped me, and of course, acknowledgements to the Bioconductors, developers, and users that created the packages that I'm running inside the omics shield. And that would be all.
Thank you very much. Questions on federated privacy-aware studies?
Thanks a million. Does this, so just trying to understand how Opal and Data Shield work, do they also work with, say, things coming out of like Beacon and the EGA, or alternatively with stuff that Odyssey and OMAP are doing?
At the moment, I have developed methods to connect to the EGA database. So if you have a user with access to some sort of data set, it can be included inside the ecosystem. And Beacon is not implemented yet, but it could be very easily integrated.
Okay, thank you very much. I would like to welcome our next speaker, Junyan Lu, who is a former researcher at the EMBL and now leads a computational omics group in Heidelberg. And he will talk about the omics studies and data integration in the context of cancer studies now. So welcome.
Thank you for the introduction. So welcome to my talk. So today I'm going to present you a method also implemented a bioconductor package called DEP-infer, which can be used for inferring tumor-specific cancer dependencies through integrating ex-vivo drug screening data and drug target profiling data. So why do we care about the cancer dependency? So as many of you may know, the survival and sustained growth of cancer cells normally depends on a handful of pathways. So those pathways, the activity of those pathways can be considered as Achilles heels of the cancers. So therefore, by targeting those pathways, we can really kill the cancer cells. But those pathways, the dependence of those pathways varies a lot in different cancer types and even among patients with the same cancer type. So that's why we need to identify a patient or tumor-specific cancer dependencies. Currently, there are already quite a lot of projects focusing on this issue. For example, this StepMap project from Broad Institute trying to identify the cancer dependencies using cell line screens. But the one problem is that most of such studies are focused on cell lines. For better clinical translation, we actually need to identify such cancer dependency in a patient or tumor tissue specific way. So currently there are mainly two ways to identify cancer dependency. One is using genetic perturbation screen, like the widely used RNA screen or CRISPR-Cas9 screen. So the advantage is quite obvious. So through this type of screen, you can directly get a list of the essential genes. So those genes are easy to interpret, so they can be the drug targets. However, there are also several issues with this type of genomic screens. The first is that the knockout of the gene is not always the same as the inhibition of the enzyme product of the gene, because the enzyme, the inhibitors can inhibit the enzymatic activity, but leaves the scaffold activity of the protein intact. And also this type of screen is not so easy to be performed on patient tissues, tumor samples, especially for some tumor cells that does not grow very fast. And also the genes you identified are not always targetable. So the other way for identifying such cancer dependency is using chemical perturbation screen, such as drug sensitivity screen. So also there are quite a few advantages, like it's easy to apply to patient samples, and also it can directly read out of the drug effect, and the targeted, the proteins or dependent genes you identified are always targetable. However, there's one big issue is that many of the drugs have multiple targets. This leads to this unspecificity of the drug target leads to the results, leads to the difficulties in interpreting the result. So we're wondering how to combine the best of the two worlds. Therefore, we came up with a method called depth-infer cancer dependency inference. We're trying to combine the information from a drug target profiling and a drug screen. So the input are just two matrices. One matrix describe the binding affinities of the drugs to different proteins. So this matrix can be from typically like a kinase inhibitor screen, like the Keynote screen, or also it can be from a thermal proteomics profiling screen, just to describe how strong the drug can bind to different types of proteins. The other one is cell viability screen after drug treatment. It can be on cell lines, on patient samples. Then we were using a multivariate, multi-target linear regression model with a Lassupinoti to estimate a coefficient matrix to explain this drug viability matrix by using the drug target profiling matrix. So this coefficient matrix can be considered as some kind of a protein dependence matrix. And the advantage of this method is that this protein dependence matrix resembles the essentiality score in this RNA CRISPR screen, but with closer relation to drug effect. Therefore, the interpretability of this method is higher, although it uses drug screening data, but it can directly tell you which protein or which gene may be important for the survival of the cancer. And also, this can infer patient tumor-specific vulnerability of the cancers. So because the screen can be easily applied to patient samples, and also the identified the targets or the proteins are always targetable because they're already targeted by the drugs. So this is a typical output of this step infer. So the output is a matrix describing the dependence of each proteins in each cancer cell line or patient sample. So here I use this GDSC drug screening data as an example. So I use this as an input and got this protein dependence matrix. So the more red means the sample or the cell lines depends more on this protein, while if it's blue, it means inhibiting this protein may lead to even better survival of the cells. So what we can do is that we can correlate this protein dependence matrix with different types of cancers. And also we found that the correlation makes quite a lot of sense. We found that the HER2 positive breast cancer cell lines have higher dependence on the HFR kinase receptor. So this is already known. And also some blood cancer like the DLBCL or B-cell lymphoma also has higher dependence on the BCR-ABL pathway. So that captures the known dependence in the cancer cell lines. And also we can correlate this protein dependence matrix with different genomic markers in those cell lines. And also we can identify like genomic gene mutation specific dependencies. Like we found that the NRAS mutation increased the dependency of the MAP2 kinase. So this is also known. So our method can capture the known dependencies of cancer cell lines and related genomics. And also one advantage is that our method can be applied to the screening result from primary tumor samples. So we also applied it to our in-house screening data set and chronic lymphocytic leukemia. So we also get this protein dependence matrix. We also found that this protein dependence matrix inferred by our method can cluster basically the subtype of the cells better than using the drug response data alone, and can also capture the known genomic associations between the kinase dependence in the CLL cells. And also we also show that our method can actually identify some novel kinase dependency in CLL. Through our method, we found that actually the CHEK1 kinase, the dependence of the CHEK1 kinase is higher in the subtype of CLL without this IGHV mutation. Although we already observed that the UCL cells have higher sensitivity to the CHEK1 inhibitors, but at that time we didn't know whether this is due to the off-target effect or the on-target effect, because the CHEK1 inhibitor has a lot of target. But through our inference, we are more sure that this may actually just through the untargeted effect, that this CHEK1 kinase is more important in IGHV mutated cells. So this also supported by some recent publications that indeed the CHEK1 kinase 1 is essential for the growth of normal B cells and also for lymphomagenesis. And also using other omics, we also show that the pathways related to CHEK1 kinase, like the checkpoint pathway and DNA repair, are highly upregulated in the UCL subtype. So our method has implemented as a bioconductor package, and so with nice documentation, and show you some examples on how to use it. So the take-home message, so we have devised a computational method for identifying tumor-specific cancer dependence through integrating drug target profiling and drug sensitivity screen data. And also our method, I showed you that our method recapitulated the known kinase dependencies in cancers and the primary tumor samples, also identified a novel association between cancer dependence and cancer subtype. And we implemented our method as well documented our bioconductor package called DefInfer. So yeah, so last but not least, I would like to thank all the people who have been involved in this project, especially Alina, who was former master student. Now she's a PhD student in EMBO Barcelona, and she did most of the work. And also some special thanks to Peter, who helped us review the bioconductor package and helped us a lot to improve the package and the coding. And of course, thank you for your attention.
Thank you very much. Questions? Maybe I can ask one, are you taking advantage on these different multi-assay data containers that are available in the bioconductor system, and do you support them?
Yeah, currently we are not using the multi-assay container. So during the package review process, Peter asked us if we can use some bioconductor containers, but actually I find that just using the matrix format is easier, less complicated for the users.
Okay, thank you. Other questions? There is one.
Thank you for your talk. It was really enlightening. I have a question about the datasets. Are they like published somewhere, or is there any consortium generating these drug sensitivity assays in a high throughput manner? Because what I see is that the method is really powerful, but I find it quite difficult to obtain the data in it. Thank you.
So for the drug protein profiling data, we're using currently one data set generated from a group in Munich. So it's using Kinobase to profile the targets of maybe several hundred of drugs and their kinase targets. And for drug screening, actually, there are a lot of available data set like this GDSC I mentioned, and also from Broad Institute, they also have their drug cancer cell line screening. But for primary data, so you can generate your own data, like collaborate from collaborators, and also there's some online for each type of cancer. Maybe there are already some drug screening data available. Like at least for CLL, we generate a lot of screening data, and also I know for AML there are also some drug screening data.
Okay, thank you. Maybe one last question.
This is kind of on to the last question. Do you have links into the pharmacogenetics frameworks and the packages that they've developed, which mostly include kind of transcriptomic data?
Yeah, I think it's a good idea. So maybe in the future we can work some way like connect those two packages and also directly query information from their package. So also we had to look at this, but it seems this this package looks quite complicated. So I need some more time to learn more about that. Yeah.
Okay, thank you very much. Thanks for all the speakers once more. And yes. And sorry, we went 3 minutes over time. It's on me. So we will have now 1 hour, a little more than 1 hour for lunch. So we continue, I think, 1:25. Be back here then. Thank you, and good lunch for everybody.